right. Well, hello everyone. Welcome back for those who have seen no neuropsychology lectures before. And if you are new, thank you for coming. My name is Dr. Casey Hewitt, and for the summer, Annie Reyes and I will be your moderators. We are pleased that you could join us for this week's lecture in the first 7-week neuroanatomy mini-series that brings you lectures from experts in the field covering different neuroanatomy topics each week. The series was created by trainees and early career neuropsychologists to provide free, high-quality didactic opportunities. We also would like to recognize the No Neuroanatomy Planning Group for their hard work for pulling this mini-series off. We would also like to thank our sponsors for their financial support for the series. And we also want to say that before we start, here are our disclaimers for the series. Training is not meant to replace any formal education in neuropsychology, and the views of the speakers are their own. Questions can be submitted through the Q&A box in the lower left side of your screen, and a recording of today's lecture will be provided on the website later this week. Today's Q&A is led by Annie Reyes. Now it is my pleasure to introduce Dr. Christopher Benjamin for today's lecture titled Introduction to Neuroanatomy. Dr. Benjamin is a clinical neuropsychologist and assistant professor in the departments of neurology, neurosurgery, psychology, and radiology at Yale. He completed his PhD training in neuropsychology at the University of Melbourne, Australia, research fellowship at Boston Children's Hospital and Harvard Medical School, Boston, and his clinical fellowship at the Smell Institute at the University of California, Los Angeles, where he was an epilepsy fellow and chief fellow in neuropsychology. As a clinician, he's a member of Yale's Comprehensive Epilepsy Center, and he also sees adults with a range of neurological disorders. As a researcher, he focuses his work on fMRI and cognitive assessment to predict outcomes of neurosurgery and epilepsy.
Okay. Thank you very much for the opportunity to speak today. So neuroanatomy is a really fascinating topic. I think it's one of the most interesting areas that we learn about. Obviously, the brain is just an area, something that's incredibly fascinating to us all. It's also one of the most difficult, or one of the most sort of tricky areas. I say this not to discourage you, but to really mention the fact that today you'll hear a lot of information today, and you shouldn't be overwhelmed by it. You should really sort of treat it as like a bit of a sort of tasting course to sort of get the main structures of the brain, to really get some ideas about different functions the different areas support, and also to get some ideas about areas that you may want to go on and learn about subsequently. The other real purpose of today is to give you some key basics so that you can make the best of the subsequent seminars that you're going to have. So while I'm providing you this introduction, hopefully you'll then sort of get all of the basics of the different lobes of the brain, the different hemispheres, and then make the most of the subsequent talks that will be provided over the coming weeks. So, as we've noted, the key hemispheric and lobar functions, the key nomenclature, the way to sort of navigate the brain. And I wanted to highlight a particularly useful text for everybody. It's by Hal Blumenfeld. There's actually just been a new edition released, and it's Neuroanatomy through Clinical Cases. Many of you will already know of this. It's a particularly good text because Hal has a wonderful way of communicating concepts. It's very straightforward, while there is a lot of detail, it tends to be reasonably simple. And he illustrates, as the name suggests, concepts with clinical cases. So it tends to be particularly engaging. I've tried to do a little bit of that, take a leaf from his book today and, as it were, and provide some clinical cases as we go. I've also really based this talk on the introductory chapter from this text. That's very deliberate because if you are anything like me, when you start out learning neuroanatomy, you really are not going to capture everything, and you will want to go back and pick up more on certain areas, and that will allow you to do this. And the slides are available afterwards for anybody who would like them subsequently. I also wanted to mention that because it's a complex area with a lot of rote learning, one of the best things you can do to familiarize yourself with neuroanatomy is download one of the many good neuroanatomy applications that are available for your phone, on the iPhone or Android or wherever. And whenever you get 5 minutes, when you're on the bus, when you're having a coffee or whatever, just flip through some cards and sort of refresh the key structures and the key landmarks, and then you'll gradually learn these over time, of course. The other thing that's extremely helpful is if you take the time to, if you can, take the time to be involved in a research study where you do something like neuroimaging, and you then get to really explore the brain sort of hands-on, as it were, with software that lets you look through the brain in detail. So starting off with some terminology here, the terminology that's used, there are a couple of different sets of terms that are used to orient you within the brain. So as you go forward in the other seminars, you're going to be hearing the speakers talk about areas that are more dorsal or ventral to one region or another, more rostral or caudal. And this, if we go back to sort of look at a reptile, you can notice that in more basic animals, the central nervous system is essentially linear, or it sort of propagates along a single axis. And so the significance of this is that here we've got really just 4 coordinates that we can use, and that will, sorry, 4 not coordinates, 4 terms we can use, and they can be used consistently at any point throughout the central nervous system of this animal. So to make things complex, everything is named with reference to Latin terms. Dorsal, we've got the back here. So dorsal, you can think of a shark's dorsal fin, of course. Ventral comes from, I think, the Latin root ventra, which refers to the stomach. And then rostral actually refers to a beak. So rostral is sort of towards the front, and then caudal referring to the tail here. So you can at any point here say that, define an area in these terms. So for instance, this area of the central nervous system here in the lizard is rostral to this area back here, whereas this area back here is caudal to that region there. When we step across to the human, we've got some complexity because the central nervous system in the human is actually not straight, it actually curves. So in the brainstem, it essentially does what you might think of as a right angle turn and heads down the spine. The significance there is that while you can use these terms dorsal, ventral, rostral, and caudal, they'll mean different things at different points throughout the central nervous system. So in the brain, if we step forward, in the brain here, in the cerebral cortex, we've got dorsal at the top, which is we can also refer to as being superior. We've got ventral down here, we've got rostral, and we've got caudal. Now, when we move down to the brainstem here, we've done that right angle turn, and what happens is we essentially have to rotate these terms. So now dorsal refers to the back rather than to the top. Ventral is now the front rather than the underneath. Okay, so these terms, dorsal, rostral, caudal, and ventral, they are relative to the area where you are in the central nervous system. You've got a separate set of terms, superior, inferior, anterior, and posterior, and they are always fixed. They will not change regardless of where you are. So the front of the brain here is an anterior region, the back here is a posterior region. Similarly, when we go down to the brainstem and to the spine, the back is posterior and the front is anterior. Some additional key terminology refers to medial and lateral. And these are again, these are somewhat relative terms. So you can describe one structure as being more medial or more lateral than another. So what you've got here is one hemisphere of the brain, and this is the lateral surface, the outer surface. And here down the middle is the medial surface. And if we bring up the other hemisphere as well, you can see that these terms are constant on the 2 hemispheres. You've got the lateral surface on the outside of each hemisphere, and then you've got the medial surface down the center here. Two other terms that are particularly important are ipsilateral and contralateral. Ipsilateral and contralateral. So ipsilateral refers to something being on the same side, and contralateral to being on the opposite side. Okay. So contralateral on the opposite side. Sorry, I just saw a hand go up there. I'm not able to manage that function there. So if any, I don't know, Casey, if you're able to jump in if there's anything that I should address as we go. Otherwise, I'll keep going as we're going. So ipsilateral on the same side, contralateral on the opposite side. So if I said to you that this hemisphere on the left controls the contralateral side of the body, I'd be saying that this left hemisphere controls the right side of the body. If instead I said to you that a small number of motor fibers that leave the left hemisphere control the ipsilateral side, it'd mean that those small amount of fibers are controlling the left side of the body. The next thing to do is to orient you to how you can understand different sections that you see of the brain in different settings. So there are three different orthogonal planes that we look at with the brain. There's the horizontal plane, the coronal plane, and the sagittal plane. The horizontal plane you can think about in terms of looking at the horizon. It's a flat plane going directly through the brain. This is also termed the axial plane or a transverse section. And so as you go forward with the other talks, you'll hear speakers talking about, so, looking at these coronal sections, we can see this structure well, or this structure doesn't turn up well here, but we can see it well in the sagittal plane. They're referencing these different coordinate frames here. So again, the horizon indicating the sort of horizontal plane. The coronal plane refers to, the word corona sort of refers to a crown, and you can think of a tiara-like crown. And so such a crown goes down like this. That section is cutting straight down through the head in this way. Sagittal plane comes from the root of the word, the word sagittal. The root of the word in Latin refers to an arrow. And so the sagittal plane you can think of as being, if an arrow was shot directly straight through the brain, it's a section that just goes straight down the center of the brain. So here we've got an MRI image down the bottom, and we've got these terms up the top. This is, in this image, the very front of the brain is up the top here. The eyes are further down. You can't see them on this single slice. This is a horizontal plane. This is a coronal plane, so just cutting down like so. And then finally, this is a sagittal plane running through the center of the brain, and you can see a number of structures there. At this point, you should be confused about what those different images are showing, and you should not be able to make a tremendous amount of sense of them if you're a normal individual early in your training. So don't be put off by that. Okay. So with respect to the central nervous system and the peripheral nervous system, what we care about to a large degree, or sort of mostly in neuropsych, is the central nervous system, the brain and the spinal cord. There is a separate peripheral nervous system which extends beyond this, and peripheral nerves are involved in things like, well, they're involved in really most functions that we are involved in. But if you, for instance, want to move a limb, once essentially the information or command has been passed down to the spinal cord, it then leaves the central nervous system and then runs out through the peripheral nerves which will then innervate a muscle, and which will then contract and allow you to move your arm. On the other hand, if you are actually feeling something, information will be detected by peripheral nerves and then run into the central nervous system and up to the brain for further processing in most circumstances. Now, a set of concepts can be particularly useful when you are trying to think about how the brain is structured and sort of what all of these different terms mean and how different parts of the brain interact with one another. So the central nervous system originally arises from the neural tube, and it's essentially a sheet of cells that start off forming what's called the neural tube. You can see here that there are a number of different outpouchings that are running through this tube here. And this tube is filled with CSF. It's filled with a fluid. CSF I'll talk a little more about later, but it's a substance which carries a lot of vital nutrients for the brain. It takes away a lot of substances that need to be removed from the brain. And you can see here, this is a particularly useful schematic because it breaks apart the forebrain, the midbrain, and the hindbrain. The forebrain is what will end up being the cerebral hemispheres. You can see down here on the right side, the cerebral hemispheres here, the major structures of the brain that you look at, especially when you're looking at the brain from the outside. In addition, it includes the grey matter on the outside, the white matter underneath that, and then within the actual, within each hemisphere, there are a number of grey matter regions in the centre of the brain, and I'll talk more about grey and white matter in a moment. Okay, so just briefly, when we look at neurons, neurons are, of course, the key cells that we have in the brain. And what you've got here is a reasonably simple model of a neuron. It may not look simple, but this is a relatively simple model of a neuron. So here we've got the body of the neuron, and then it's receiving input in the most basic sense. It's receiving input from a number of different processes that are coming off it here, which are referred to as dendrites. And so most neurons in the body will have, in the brain rather, will have a large number of dendrites. And the dendrites, as I'll describe in a moment, are collecting information in a sense from other neurons out the brain, throughout the brain. The input that's coming from other different neurons can be excitatory or inhibitory, and that just means that it can push this neuron towards a state of excitation or a state of inhibition. So it can make it more likely that this neuron will actually fire and sort of communicate information with subsequent neurons, or make it less likely, be excited or inhibited. So excitation and inhibition, the balance of excitation and inhibition, is determined by all of these inputs to the neuron. So this cell body has got a large number of other neurons passing on voltage to it and causing changes in the overall voltage in this neuron. And when a critical level is reached, there's a critical level of excitation that's pushed into the neuron. What's called an action potential is triggered. When an action potential occurs, there's it begins here, and a wave at the very start of what is referred to as the axon, and the current then propagates through this output process, the axon, all the way down to a series of synapses at the very end. And you can appreciate that there's a single axon to start with, and then there are a number of different collateral axons here. We used to historically think that there was a single axon of most neurons. We now know that most neurons actually give multiple different axons. So at the synapse here, what happens is that a neurotransmitter is released. The neurotransmitter is then taken up by the dendrites of the subsequent or the post-synaptic neuron, the next neuron, and the effect of that is to then change the voltage in the subsequent neuron, which may then lead to that other cell firing. Okay, because we have a relatively limited time, I'll keep going from here, but there's a tremendous amount that could be said about this, and you can get a great deal from Dr. Blumenfeld's book there. So to step across to the macroscopic level, we can see that if we take a slice from this brain here, we can see a very, very large number of neurons. And you might think to yourself, first of all, what slice is this? So use this to try and sort of refresh your memory and learn as we're going. Is this slice a horizontal slice? Is it a coronal slice? Is it a sagittal slice? So horizontal, think of the horizon. We've got a flat line like that. this is not being cut in that plane. Coronal, we've got the crown, the tiara cutting down. It's a coronal slice there. Okay. So what you're going to appreciate when you look at this coronal slice is that around the outside of the brain, there's a whole lot of gray matter. And this is arbitrarily colored gray, but it is actually also gray in the substance of the brain. But on different, I say that because on different imaging, it can turn up as different colors. So this outer layer here on the surface of the brain is the cerebral cortex. And this is predominantly the cell bodies or the processing components of these neurons. And so you've got a very large number of neurons throughout the cortex, and neurons are then communicating between each other, between themselves, with each other throughout the cortex. So there are a whole lot of axons running. There are axons that are actually in the grey matter, and also there are a large number that sort of dip down out of the grey matter into this white region. And then separately, there's a lot of long-term communication, or rather long-distance communication, that is supported by axons running through this white matter. So you've got neurons that are here, they'll send axons across to the other side of the brain, and also all throughout the brain within the same hemisphere, and in the contralateral hemisphere. The contralateral, again, contra being opposite, the other side, the other hemisphere. Okay. The other thing that you can appreciate here are gyri and sulci, or gyri and sulci. So the sulci are these little areas dipping down, and the gyri or the gyri are these areas that protrude out from the brain. So this convoluted surface that we have is constituting these gyri going up and the sulci going down. And there are a number of different theories as to why this high level of convolution occurs. There's the suggestion that because it creates so much more surface area, it allows for more cell bodies there and greater processing, for instance. And there are a lot of neuroimaging methods that have developed over the last 10 or 20 years, which will actually accommodate this. So you sometimes see maps of the brain where the brain's actually just it looks like a sphere, looks like a balloon, because in some cases now you can analyze the data more effectively if you sort of spread the cortex out like that. The other thing you can appreciate here is that in addition to the grey matter on the outside and the white matter on the inside, there are also some areas that you can sort of just see here. These are groups of neurons that are within the hemispheres themselves. So they're sort of almost little islands within the white matter, or structures within the white matter. And these are referred to as nuclei. So when you hear someone talk about deep subcortical nuclei, for instance, what does that mean? Deep, just far down, of course. Subcortical, below the cortex. Nuclei, groups of neurons. So there are a large number of grey matter structures throughout here, which we'll talk about in some detail. The thalamus, the basal ganglia, for instance. Another thing you can appreciate here is the corpus callosum, a large white matter structure that runs between the two hemispheres. You might remember the corpus callosum from experiments that you've heard about completed by Gazania and colleagues over time. In the split brain studies, the corpus callosum was sectioned in patients, and it was then no longer possible for the left and right hemispheres to communicate effectively. That's rarely done these days, although it does happen sometimes, for instance, in some forms of epilepsy. In epilepsy, seizures occur because there's a small area of the brain which is essentially malfunctioning, and sometimes people will actually fall down and be very disabled or incapacitated purely because, or largely because, that seizure is spreading to the other hemisphere. So if you can actually section the corpus callosum, you can stop that seizure spreading and help the person that way. Now, a key distinction that I wanted to note for you is the distinction between the left hemisphere and the right hemisphere. There are a large number of distinctions that are made. For instance, there's the idea of a more creative hemisphere or a more analytical hemisphere. Those are really very, very gross simplifications, and in a lot of ways, they're really not that helpful. Perhaps the most helpful distinction that can be made is between the left hemisphere being more involved in language and the right hemisphere being more involved in spatial function. You've noticed that I've said more there because it's really not a simple binary thing. For instance, we know that while the left hemisphere is involved in most aspects of language, the right hemisphere actually supports some functions like, for instance, prosody, or the sort of sing-song nature of speech when you're talking. And similarly with right hemisphere function, we know that while areas of the right hemisphere are more involved for a large number of spatial functions, it's also the case that some aspects of spatial function are performed bilaterally. So, for instance, areas around in the middle of the temporal lobes are often involved bilaterally in some aspects of spatial function. So I just wanted to give you an idea of what somebody who has an injury in the left hemisphere might look like. So this is an individual who's had an injury just in the left hemisphere.
Hi, Byron, how are you?
I'm happy. Are you pretty? You look good.
What are you doing today?
We stayed with the water over here at the moment and talk with the people, with them over there. They're diving for them at the moment. They'll save in the moment. He had water first, and for him, with luck for him.
So we're on a cruise and we're about to
We
get to
will
Juneau.
sort right here, and they'll save their hands right there for them.
And what were we just doing with the iPad?
Right at the moment, they don't show a darn thing.
The iPad that we were doing. Like here?
I might change for me and change hands for me. It would happen. I would talk with Donna sometimes. We're all with them. Other people are working with them. I'm very happy with them.
Good.
This girl was fairly good and happy when I play golf and hit other trees. We play out with hands. We save a lot of hands on hold for us, other hands. I don't know what you get, but I talk with a lot of hand for hand.
So it's this striking video, isn't it? That's an individual with a Wernicke's type aphasia, and this is the left hemisphere. This is a wonderful paper by Jeff Binder from 2015, outlining key areas of the language system. And there's an area in the posterior temporal gyrus up to the parietal cortex, which is particularly involved in pulling out the sounds of speech. And this individual hasn't had an injury there. You can see that he is able to still speak. He's still able to make sounds. He's actually still able to say words, but he's not accurately comprehending what's been said to him. She'll say something about the iPad to him, and he looks out the window, I think it was, and looks as if he's thinking about something else. And he also can't actually select the right sounds that he wants for the concepts that he's providing. So he's saying things for all intents and purposes, but he is saying what he is actually saying is not reflecting what he is thinking. And then in addition, he's actually not actually able to comprehend what he is actually saying. He's not hearing that there's a disjunct between what he intends to say and what is actually said. More anteriorly, so what does anterior mean? Remember, anterior at the front, and the eyes would be just down here, posterior at the back. More anteriorly, there's a region that's just involved in perceiving the sounds of speech. And unlike this posterior region, more area towards the back of the brain, which is involved just on the left hemisphere, really, in processing the sounds of speech, pulling out representations of them that are actually kept in the brain. This area at the front, which is involved in just perceiving them from auditory input that you receive, is actually bilateral. I didn't introduce it as bilateral, but bilateral is both hemispheres. And there's an amazing phenomenon that's very rare. When you get a lesion to both of these regions that are yellow in both hemispheres, you actually can get something called pure word deafness. And what happens there is that people actually are not able to hear the sounds of speech. So you could hear this call of a bird and know what was happening. You could hear the sound of a car and know it was a car, but when people talk, you just can't hear what is said. But you are actually able to retrieve the sounds of speech and speak yourself. It's just that you can't hear or perceive speech sounds. So on the other hand, the right hemisphere is particularly involved in spatial function. And this is a particularly good video as well. And I should point out, these videos are already in the public domain. I wouldn't be just showing you videos of a patient who we had seen or someone else had seen, of course, because there are all kinds of ethical issues associated with that. But these videos are on YouTube, and the references are actually in the slides here. So this is a particularly good video, not only because it's Australian.
Put the numbers on the...
Oh, here they go.
Yeah, where the numbers go.
1. Is that how it should go? 2.
Yep.
3. 4. 5. 6. 7. 8. 9. It's my birthday yesterday. 10, 11.
Yeah. That's good. How many got 2 trying to go?
Okay, so you can see there that she's trying to draw a clock face, and I should say you can see there, I apologize for the fact that it's not a fantastic video, but it's actually hard to find a video of something like this. So while she's trying to draw a standard clock face, you can see that she has essentially neglected the left side of space there. She's just drawn the numbers down this side, and she's tried to cram all 12 of them, and you might have heard at the end there that she stopped and she checked her work and was like, is that correct? And eventually concluded that yes, it was. So what you're seeing here is an example of neglect after injury in the right hemisphere. And so neglect can occur after injury to a large number of different regions. And these are the results of a meta-analysis by Jason Mattingly or Jason Mattingly's group, I should say. The primary author was Molenberg. And they show that there are a number of different regions throughout the right hemisphere which lead to, or which can be associated with neglect. Most commonly, the region associated was here in white matter underlying the parieto-occipital cortex. Don't worry if you don't understand that, we'll talk about the lobes in a moment, but the significant thing here is that lesions in the right hemisphere, a lesion being an area of damage, areas of damage in the right hemisphere can lead to neglect like this. We also now know that there are a whole lot of sort of different flavors of neglect, or different types of neglect that you can see on different tasks. Like you might see one form of neglect when a patient in day-to-day life is not, they sit down and see their meal and they only eat the food from the left side and don't eat the food from the right. And sometimes patients with those deficits don't necessarily have deficits on some of the testing we're doing. There are a large number of tests we do, and some forms of lesions in some areas of the right hemisphere will cause deficits in some of those daily tasks, or on some of the tests that we do, and others will cause deficits in other areas. So, and there's a question about the source of the Wernicke's video. The sources are actually in the slides, so you'll be able to access those afterwards. But if you just go to YouTube and Google, and search for Wernicke's fascia, you'll be able to find that there. So what you've got here is the major structures, the major lobes rather of the brain. And so I'm going to very briefly introduce you to these lobes and what some of the key functions of them are. So at the front of the brain, we'd have which is here, we'd have the eyes sort of essentially down here. And this is the back of the brain here. So here we've got in blue, the frontal lobe. And this is very arbitrarily named as Bob Bilder pointed out at one point. We could just refer to this section back here as the basal lobe, but we've arbitrarily decided to call this the frontal lobe because it's towards the front. So the frontal lobe is involved in, well, actually, I should give you the way to sort of determine where the frontal lobe is, because of course these colors have just been added here. There's a long sulcus. You remember a long sort of valley that runs down the side of the brain here that's called the central sulcus. So you can identify this because it's particularly large and it runs all the way right down to this area here. This is a second major landmark on the lateral surface of the brain, or the outer surface of the brain. It's called the Sylvian fissure along here. So the frontal lobe is bounded by the central sulcus down here, and then the Sylvian fissure down here. So I could also say, of course, it's bounded by the central sulcus posteriorly at the back, and the Sylvian fissure inferiorly down the bottom down here. The frontal lobe is particularly involved in a large number of functions. It's very interesting because historically, injuries to the frontal lobe often didn't correspond to injuries on the kinds of tests that we do. You see things like changes to personality from injuries to the frontal lobe. The skull at the base of the frontal lobe is actually it's very convoluted. There are a large number of bumps that come up out of the skull. And so when you do something like have a car accident, have a fall down a set of stairs, as the head shakes, the brain moves around. And unfortunately, that's an area where you can get the brain being damaged by its movement along the skull there. And so you can actually get changes to personality from injury, in particular to the orbitofrontal regions at the front of the frontal cortex there. The dorsolateral regions, which are dorsal, so towards the back, and we're in the brain, so it's up the top, lateral towards the outer surface. The dorsolateral cortex here is particularly involved in working memory. We've got an area down here, Broca's area rather, which is really involved in language as well, in production of language. Saying production of language is a gross oversimplification, but it's very involved in language. There's a really interesting area here towards the back of the frontal lobe that's also really involved in handwriting, and I'll talk more about that if we have time as well. Here in green, we've got the temporal lobe. The temporal lobe is particularly involved in language and in memory. It's involved in many functions, but they're two of its key functions. So while the frontal lobe is involved in things like personality and in working memory and language, and overall, it's a large group of functions other than language that we call executive functions, and many of you will have heard that term before. In contrast to that, the temporal lobe is involved in language and memory functions. So there's an area towards the center of the brain, the hippocampus, which is a key memory structure. And so if you have damage, very severe damage to the hippocampus within both hemispheres, you lose the ability to retain memories over time, retain new memories. And so many of you will have heard of the case of HM, who lost both of his hippocampi due to a surgical procedure. It was an exploratory surgery. It was done with the patient's consent a long time ago, and was done to try and cure a condition which could have led to his death in the long term. So something like that is never done lightly. But if you have damage to the hippocampi in both hemispheres, you can essentially become amnesic or have great difficulty forming new memories. If you have injury in one hemisphere, you can still have memory difficulties. In a posterior region of the temporal lobe, right at the back here, there's an area that's called the basal temporal language area. And you don't need to remember the names of these areas. These are just for your interest. As long as you remember the temporal lobes involved in things like memory and language, that's going to be sufficient to get started. But the basal temporal language area is really involved in retrieving the names of different objects. So when I look at the computer screen now and I say, oh, that's a screen, that area is involved in associating the image of a screen with the word screen. So in terms of the boundaries of the temporal lobe, the temporal lobe is bounded superiorly towards the top by the Sylvian fissure, and then inferiorly it's just the base of the brain. And then posteriorly, there's not really a good sort of boundary that demarcates the end of the temporal lobe. And so there is a sulcus that runs down the side here, and there's just a line drawn between this sort of little notch this is the parieto-occipital sulcus, it's called, between the parieto-occipital sulcus and the back of the sylvian fissure there. So this is a somewhat arbitrary line that's drawn here. So if we move back to the parietal lobe, the parietal lobe is involved in a large number of processes as well. In the left hemisphere, it's particularly involved in language function. In the right hemisphere, it's particularly involved in spatial function. So in the left hemisphere, there's an area, Wernicke's area is sort of at the back here, and there's also an area around this lower region of the parietal lobe, which is involved in going between written forms of language and spoken forms of language. This area is also very involved in calculation in the adult. Calculation is another one of the many interesting areas. When we're young, some aspects of mathematics and calculation tend to be done by both hemispheres bilaterally, tend to be done bilaterally. And then as we become more proficient at them and more skilled, complex language processing tends to be done unilaterally in one hemisphere, and it tends to be the language dominant hemisphere. So that's not really strong, it's not as strong a relationship as some of the other relationships we see, like language to the left hemisphere, but this area does tend to be involved in calculation and math in adults, or some aspects of that. On the other hand, I mentioned in the right hemisphere, lesions in this area are particularly associated with neglect. We've got the occipital lobe back here. The occipital lobe is really involved in vision. So while we've got the eyes at the front here, there is the neurons of the eyes send white matter back into the brain, and it ends up traveling all the way to the very back, and the back of the brain is involved in, or cortex rather, at the back of the brain is particularly involved in processing visual information. There are also a large number of extremely cool visual areas that proceed forward from the occipital lobe. So one of the many features of the brain is that as you move away from primary cortex is an area which processes sensory information first, or processes output from the brain last. So in the case of the visual system, the occipital cortex is primary because it's where the first processing, or the first cortical processing of that information is. As you move away from there, the representations in the brain get more and more complex. So within the cerebral cortex in the occipital lobe, you might just have representations of very basic information, like a very sort of crude, unprocessed information about what's being perceived in space. As you move forward, you have specific areas that are selectively involved in processing things like form, that are involved in processing color. So you can get lesions in around here, this junction between the occipital cortex, the parietal and the temporal cortex. You can get injuries that will selectively knock out the ability to perceive color, a chromatopsia. You can also get a selective inability to properly perceive movement from injuries in some areas. And underneath the temporal lobe and occipital lobe moving forward, there are also areas that are selectively involved in processing things like letter forms. There's an area called the visual word form area. And so you've received basic visual information, it's been processed more and more moving forward. And then in this area, the visual word form area, it selectively responds to word forms. And there's some great work by Nancy Kanwisher at MIT and others, which has shown that this area becomes specialized over time. So for instance, if we took a Hebrew speaker and an English speaker, in the Hebrew speaker, this region would respond selectively to Hebrew text and not to English language text, and vice versa. In the English language speaker, it would respond to English language text, but not Hebrew text. So just to step through these again, so you can be thinking about these general ideas, general concepts as you go forward. The frontal lobe really involved in executive functions, and I also mentioned language there as well. The temporal lobe really involved in language, and in particular in memory, especially in an area you can't see there, but in towards the center of the brain there. The occipital lobe really being involved in vision. And then we've got the parietal lobe, which is involved in many functions, but in the left hemisphere, particularly in language and also calculation I mentioned. In the right hemisphere, it's really involved in spatial function. And in relation to that, it's also involved in things like proprioception, the ability to know where your limbs are in space in the right hemisphere as well. Okay, now I'm going to skip ahead just a little here, unfortunately, as we don't have as much time as I had planned for. And I'll try and show you some of the more interesting things that I'd put in here as well. So in relation to primary cortex that I mentioned to you, this slide captures a really fundamental concept, or captures that fundamental concept in a really nice way. So again, all of these images are from Dr. Blumenfeld's book, and I'll put the reference up to that again at the end. But I mentioned that here at the very end of the occipital lobe, we've got this primary visual cortex here. Here in the superior temporal lobe, near the top, so near the top of the temporal lobe here, we've got in blue an area that's really involved in language processing, and in auditory processing. And auditory processing is also done really just inside that long fissure, or that long, the Sylvian fissure, that long little valley there. There's a lot of basic auditory processing that occurs in there. And then up here in blue, this long sulcus here, you might remember, is the central sulcus, so you can see that there, the central sulcus that runs down here. And so why do we care about that? We care about that again because it's the division between the frontal lobe at the front here, and then the parietal lobe just behind it. So just before you get to the central sulcus, you've got the primary motor area, the motor strip. And so this area is involved directly in stimulating your muscles throughout your body when you want to perform an action. On the other hand, just behind the central sulcus, just posterior to the central sulcus, we have what's called the post-central sulcus. And I must say that with most of the sulci and the gyri, you can work out where most of these areas are just by looking at their names. So we had that pre-central gyrus, which is just before the central sulcus, and now we've got the post-central gyrus just after the central sulcus, really involved in processing sensation. So we've got these primary areas here, and there's a primary area involved in olfaction at the base of the frontal lobe. But what I'd like to draw your attention to is that when we move beyond these primary areas, in each case, there's one of these red areas, which is a secondary processing area. So I talked about, for instance, how in the visual processing, we go from basic features to more complex things like representation of objects or letters and the like. These red areas are referred to as unimodal association areas. Unimodal association areas. The idea being that they stay within the one modality, unimodal, one mode. So the visual modality here, for instance, and they integrate aspects of or they do more complex processing on that basic visual information. Then we go to the auditory cortex inside the Sylvian fissure, and then we've got a secondary cortex just around that as well, doing that additional processing. Got the motor cortex just in front of it. We've got some secondary association cortex here. Got the post-central sensory cortex again, and we've got some secondary associative cortex there as well. And then finally in yellow, we've got what we can refer to as tertiary associative cortex, or multimodal associative cortex. So the idea there is that once you move beyond the secondary cortex, or the unimodal associative cortex, you've got areas of the brain that are pulling together information from many different areas. They're taking visual information, auditory information, other information, and associating it in even more complex ways for processing. This concept has been around forever. One of our great forefathers in neuropsychology is Alexander Luria, and he talked about these three zones, and it's a concept which is really helpful. Okay, so we're almost at the end. So one of the other really cool things I wanted to mention is that within the motor and the sensory strips, we actually have a really very organized, or they are very organized in their structure. And so what you've got here in blue and in red is the sensory cortex, and then the motor cortex. This is referred to as a homunculus, and the homunculus just means little man, and it refers to the fact that there's a very orderly representation of the body as you move through the cortex. So if we go to the motor cortex, the areas of the cortex involving the foot then progress to the areas involving the knee, involving the behind, involving the arm, the face, and so forth. So this has a number of different and really interesting consequences. One of them is that when you have a seizure or where you have a abnormal functioning in the body, you can actually, you have abnormal functioning in the brain. And please do let me know, KD, if I'm KC, if I'm running over, I need to stop. But in epileptic seizures, what happens is that there's an abnormal functioning within an area of the brain. And while the seizures most often begin in a very sort of small select area, they then tend to spread to other areas. So you can have a seizure that begins in this area of the foot, for instance, and the person's foot might begin twitching. And then as it progresses to adjacent areas, the movement will then move up the leg and then move into the trunk of the body and towards the arm and so forth. And so this process, seeing this very classic seizure where the jerking begins in one area and then moves throughout other areas, is called the Jacksonian March. This video shows it at a very basic level. I'm going to skip through it slightly because there are almost no videos of it out there, and I just wanted to give you a flavor of it because it's such a cool way to illustrate this concept. So you can see at this point, this gentleman's leg is twitching, but his foot in particular is twitching. The top of his leg is not such an issue at the moment. And as we move further through it, the twitching moves up his leg. And you can see that the muscles all through the top part of his leg now are pulling together and jerking there. And later we don't see it quite so well here, but it then moves up to the trunk of his body there. So again, the concept for you there is the fact that through the motor system here, the motor strip, this part of the motor system, I should say, the motor strip, the primary motor cortex, different areas of the body are represented next to one another. This property occurs throughout the different sensory areas and through much of the processing in the body, that areas of when information from one part of the body is transmitted to the cortex, areas from adjacent parts of the body will also be transmitted to that part of the cortex. So to finish up, I'll give you an example, a final example from the visual system. So what you can see here is, I mentioned to you that we've got the eyes at the front of the brain, and then we've got fibers that carry the information from your visual field to the cortex. This is the primary visual cortex here. And up the top here, this is just showing what the left eye and the right eye can see. So the left side of your visual space, what you can see on the left side, is transferred to the right hemisphere, the contralateral hemisphere. And these colors are just showing you where the information goes. And you can see that the area 1, we've got areas 1, 2, and 3, and that corresponds to areas 1, 2, and 3 in the visual field. So we've got this topographic organization, just like in the motor system, where the areas for the leg were next to the areas for, or the areas for the foot were next to the areas for the shin, the knee, the upper leg. Same thing within the visual system. We've got the center of the visual field, we've got the area around it, and then the periphery around that as well. So that's a general property of the visual system as well. And to give you one last example of a seizure, illustrating some of the functioning of the visual system, and you don't need to understand all of this. So this is someone having a seizure, or it's an illustration of someone having a seizure. This is what it might feel like if you were having a seizure. So you can see there that in that case, the person was having a seizure that was probably in a visual area. It may have, it's hard to say for me exactly where that could have been. It could have been in a very basic area back near the primary visual cortex, but more likely, I think it was in one of the higher order areas impacting color or movement or some aspect of form. So I've mentioned seizures a couple of times there because they do such a great job of showing what a particular area of cortex does by stimulating that area during a seizure or during a short period of abnormal activity. So thanks very much for your attention there. There's been a lot of information. Just to wrap up, remember the only things that you really need to take out of this are what are the two hemispheres specialized for? What are the names of those four lobes? Do spend a little bit of time going over those terms for navigating the hemispheres. And if you can afford $100, I know it's a lot of money, you can get Blumenfeld's wonderful book, or you can spend significantly less and get a second-hand copy of one of his older ones, which honestly, there's probably not a tremendous amount of difference in the basics between the different ones. There's a really great website called Abe Books, and you can probably pick the second-hand copy of the second edition up for $30 or something like that.
All right, well, thank you, Dr. Benjamin. This was an incredible lecture. We're getting so much positive feedback. So thank you so much for doing this. In the interest of time, we have a few questions that we thought would be very general and will apply to a lot of our attendees. One of them was in terms of what neuroanatomy application do you recommend in order for studying? If there's one, that's one of your favorites.
What neuroanatomy application?
Mm-hmm. Yes, whether it was phone app or computer-based.
Oh, right. I was trying to look this up before because I wanted to actually be able to give a really good example. And the one that I learned with has actually been removed from the App Store, which is extremely annoying. So what I'll do is I'll try and find a good example that I can provide with the slide. I'll provide a link to it with the slides to people. But having said that, if you just go to your App Store and search for neuroanatomy slides or neuroanatomy flashcards, there should be multiple free applications. So I will pass on a reference.
Perfect, thank you. Another question was, and we actually got this question from Twitter before our lecture today. If you could only read one concise review paper or chapter on neuroanatomy and brain behavior relationships, what would it be? Sounds like a tough question.
Yeah, I think I would take the chapter that this talk's based on. I'd take the chapter from Hal's book. The reason I say that is I'm not aware of any they may well be out there, but I'm not aware of any really good reviews that do such a good job of synthesizing all those fundamental concepts. Because I love language, there are references to a couple of great papers in there. So I'll just use that to plug. Jeff Binder's got this amazing paper that came out in 2015 about the language system. And I think it really changes the way that a lot of people think about Wernicke's area. So to segue into something that I'd like to offer, that's a really great paper.
Perfect, thank you. Two really quick questions. One of them was, is white matter typically thought as subcortical and gray matter as more cortical?
Yes, that's right. So yeah, that's absolutely correct. So by definition, subcortical is below the cortex. The outer layer, the gray matter, the cell bodies are definitely on the surface. Typically, I mentioned the fact that there are some nuclei that are within the brain, are further down, and then yes, the white matter sort of largely extends down from the gray cell bodies on the outside of the brain.
Great. And then last question. With pure word deafness, would one still be able to read or write language, or is perceiving any language at all impossible?
That is a really cool question. That's great work. Whoever wrote that, it's a really good way to think about these cases. And if you become a neuropsychologist, if you're not already, that's the kind of thing you need to be asking yourself when you're seeing a patient in this situation. So what you might think of there is the fact that I talked about the fact that this area involved in pure word deafness, the bilateral injuries in the superior temporal lobe, are involved in processing the sounds of speech. So yes, you can still access linguistic information. People can still speak. They can, as long as they don't have to perceive the sounds of speech, you're actually able to use language. So people are still able to speak using the rest of the brain, in particular areas like Broca's and the rest of Wernicke's more posteriorly. And in addition to that, you can also still read written language using the visual system. So you would expect, I haven't looked up pure word deafness for a long time, but I would expect someone who has pure word deafness to be able to read text without an issue, to be able to read it aloud, to read it and comprehend it, to do most other things that are involved with language. It's just purely deafness to words, an inability to hear the sounds of speech.
Thank you so much. Well, that's all we have for today, just because of the interest of time. Again, thank you so much. This was an amazing lecture and a great start to our series. Thank you everyone attending, and the lecture will be posted in a few days on our website.
Thank you all.